Okay, people, so this is going to be our biotechnology lecture, virtual lecture. I'm sorry the live session didn't work out today. I think it got overwhelmed as well because I was trying to screencast the lecture. So I'm thinking next time is going to be just fine. I've had other live sessions that worked out well. So what are we going to be talking today? As I told you guys before, we'll be looking at this technology, this biotechnology. So it's a technology where you use biological systems. We're going to be looking at electrophoresis, something that's called a recombinant technology, something that's called PCRs or polymerase chain reactions. We'll also be looking at transgenic and genetically modified organisms, cloning, and this very amazing innovative tool called CRISPR. So as I was telling you guys, recombinant technology is simply when you add DNA from a different source to another source. So this recombinant DNA is a mixture of two sources of DNA. We know that bacteria do this a lot. Okay, so bacteria have these plasmids that they can interchange amongst each other. That's also why bacteria that are, for example, not resistant to a particular antibiotic can become resistant because they can get this plasmid from another bacteria. So remember, this plasmid is this independent circular piece of DNA that they can interchange amongst each other. And we can use this information so that we can insert plasmids in bacteria, or that we can insert genes that are of interest in bacteria. Okay. When you insert a plasmid into bacteria, you need something that is called a vector, which is a plasmid carrying a new gene into a host. So usually what you can use is you can insert just the plasmid directly, or sometimes what you can use are hosts that are almost like viruses, inactive viruses that are taking in this plasmid, this piece of DNA, and injecting it into the bacteria. Okay, once you inject this plasmid into that bacteria, the bacteria can then just transcribe and translate that plasmid, creating or expressing the gene of interest. So if we want to see how this works, this is kind of how it works. You have this bacteria with its chromosome and its plasmid. You take out the plasmid, you break the plasmid, you cut the plasmid in the place that you want to insert the gene of interest. You insert the gene, and then you reinsert that plasmid into a recombinant bacteria. Now it's recombinant because it has two different sources of DNA. And then the bacteria can do one of two things. It can continue replicating that DNA, and when it replicates that DNA, it means it's replicating the gene of interest. Okay, so you can have many copies of a gene that you want to study, or it can replicate the DNA and also translate and transcribe that DNA, creating a specific protein. So if you want, for example, human growth proteins are created this way for people that have problems with their height, or protein that dissolves blood clots are also created this way. And another very important substance that's created this way is insulin. So this is biotechnology, recombinant technology, where you recombine two different sources of DNA so that you can express a particular gene of interest. How can you cut these pieces of DNA and place the DNA that you actually want, the gene of interest? You can cut them with something that's called restriction enzymes. So we believe that this was actually evolved in bacteria, and that they had this restriction enzymes, these cutting enzymes, so that they could cut the DNA from a virus so that they could not get infected by that virus. So the restriction enzyme is going to cut right over here. Okay, so it's going to leave these little sticky ends. Okay, these pink nucleotides are sticky ends. Okay, and what you can do then after you've cut the double helix, because it's not a single, it is double, right? Two different DNA segments. Sorry if that was confusing. Okay, so you're gonna cut right over here, and once you cut that, what you can do is you can insert a particular gene of interest that is going to attach right over here in these sticky ends. So then you're going to have that gene of interest be placed right over here. Okay, once you have placed the gene of interest right over there, you can insert this entire plasmid into the bacteria, and the bacteria is going to make lots of copies of that plasmid, cloning that plasmid and expressing it, right? So you're going to copy a bunch of that DNA, create an RNA, create a protein, create a trait. Okay, sometimes if we're just doing this for engineering purposes, for engineering insulin, for example, we just want right over here this protein, and then we're going to filter that protein from that bacteria, and we're going to get it to people that have diabetes, for example. Okay, so this is just a very, very brief overview. You cut the DNA, you insert the gene of interest into your plasmid, and now the plasmid is going to have that gene of interest. Of course, that when you insert something into the DNA, you need to make sure that it binds, right, to that DNA. So you're going to use the ligase in order to bind that new inserted gene into that plasmid. Okay, so this is just an overview again. Once you insert that plasmid into the bacteria, you can make the bacteria grow, and then you can harvest that protein. When you're talking about transgenic, so in that case, this bacteria right over here, that would be a transgenic bacteria because it is an organism that contains foreign DNA. So these transgenic organisms are called genetically modified organisms. What is the purpose of modifying an organism here? You can modify that bacteria to get that protein, but what you really want is a protein, right? You can also simply modify an organism so that the organism becomes stronger. So this is very important when it comes to, for example, agriculture. So you can modify a plant by giving it a gene to protect it from different pests or from different parasites. Okay, so for example, there's a corn that is genetically modified, and what it does is that you insert a gene that produces a bacterial toxin that kills a caterpillar pest. So this corn is constantly affected by these caterpillar pests. So what can we do? We genetically modify it so that it has integrated in its DNA a gene that will code for a toxin so that the pest cannot survive. You can also extend in a particular season of a plant. So, for example, fish berries. What are they? They are strawberries, but they contain an antifreeze gene from flounder, from a fish, I'm guessing. They contain an antifreeze gene. So you can grow these strawberries when the temperature is super low. So you extend the harvesting time for these the growing season for these flowers. You can also put different nutritional things into food. Okay, so for example, golden rice. It is a rice that has the ability to produce vitamin A. Okay, so you put a gene of vitamin A production into this rice as it's growing, and then when you eat the rice, you're not just eating all of the nutrients in the rice, you also eat the vitamin A that's produced by this rice. So what can you see here? When you are genetically modifying, there's been a lot of tests to do genetically modified animals, and you probably have seen images of this. They've created it's actually super easy to do. You can take a gene of bioluminescence from a particular jellyfish, and you can insert that gene into different vertebrates. And that's also how you can get these glowing fish. I don't know if you've heard about them, but they're genetically modified organisms. The fish doesn't actually have naturally the ability to bioluminesce, but the eggs of the fish are injected with this gene, and the fish inserts the gene into its own chromosome, and in that way has the ability to bioluminesce. What are some of the uses for restriction enzymes? That is one, recombinant technology. What are other uses? We can use it in forensics, in medical diagnosis, in paternity diagnosis, in evolutionary relationships. There's a bunch of things that we can do. So we can start thinking about how we figure out all of these different things. Forensics, paternity, if you have a bad allele, what are the evolutionary relationships? So you can run something that's called a gel electrophoresis. So the gel electrophoresis, of course, it's a gel. Okay, what you do is that you insert a DNA that you have previously cut with restriction enzymes. So you have cut the DNA with restriction enzymes, and that has divided the DNA into fragments based on size. Okay, so then you insert that DNA that has been cut into one of these wells. What's important about this gel is that it's actually going to have a section that is negative and a section that is positive. It's going to have an electrical charge. Okay, so right over here it's going to have a negative electrical charge, and right down here is going to have a positive electrical charge. And what that does is that we know that DNA has these phosphate groups that are negative in charge, right? So DNA wants to move towards the positive end. So since the DNA is now placed in gel, if the DNA is super thick, if the fragment is super thick and heavy, it's going to take a longer time to move because of the molecular weight of that fragment. If it is very light, meaning that it has very little amount of bases, it's going to move much faster. So you can divide this entire gel based on the length of the fragments. So the fragments that stay closer to the well are going to be the longer fragments, and the fragments that get closer to the positive end are going to be the shorter fragments. Okay, so this is kind of what it does. Negative side, positive side. DNA is trying to move, but it's kind of like swimming through jello, so the bigger the DNA fragment, the harder it is to swim. Okay. Okay, so how can we use this to test evolutionary relationships? So these restriction enzymes are cutting the DNA in particular sites. But based on evolution, we know that different fragments can differ. They can be longer, they can be shorter based on just mutations that have occurred or based on recombination that has occurred through different matings of different people. Okay, or different organisms. So all of this causes variation. So the length of the fragments are going to be compared from one organism to the other organism. If the length of the fragments or the patterns of the fragments are very similar, that means that they are super close evolutionarily. Okay, if they're very dissimilar, that means that there's been a long time since they deviated from this common ancestor. Okay, they've deviated from this common ancestor. It's been a long time, so they are pretty far away from each other in an evolutionary scale. Okay, you can also use this to see if a person has different types of alleles. So the alleles, remember, are going to be different because they're for the same gene, but they're coding for a different trait. So the allele number 1 and allele number 2 may have different sized fragments. Okay, so you can see if allele number 1 and allele number 2 are different, and you can see if a person is a homozygous, a heterozygous, and so on. You can also check if somebody is guilty of a crime, which I think is the very cool thing, is forensics using this biotechnology. So you can have in each one of these wells DNA from different samples, okay, in a crime scene. And what you do is that you cut the DNA of each of these samples with the same restriction enzyme. So you're trying to form the same types of cuts. But since some sequences may be a little bit different from the other, the length of each fragment is going to be different. It's only going to be the same if you're talking about the same person. So this is how you actually check this out. If the crime scene sample, right over here, has a small line here, and the suspect number 2 has the same line, that means that it's extremely probable that that suspect matches with the crime scene. Okay, so if we want to see it just a little bit specific, I don't know if you guys know about the O.J. Simpson case where he killed his wife. I know he killed his wife, okay, but he was acquitted and he was free for a very long time, and so whatever. So they didn't have this technology at that time. So right now what you can see is that you had blood sample 1 from the crime scene, blood sample 2 from the crime scene, blood sample 3 from the crime scene. So you had 3 different blood samples, or 3 samples. It doesn't have to be from different people. They're just 3 samples. Then you have just the standardized length that you are looking at, okay? And then you have the blood sample for the suspect, for victim 1, and for victim 2. It was Nicole Brown and another guy that died in that crime. Okay, so what you can do is then you can compare the sample of the suspect to the sample of the crime scene. And what do you see here? O.J. Simpson's sample has this little line here, 3 lines here, and 3 lines here. Okay, this is his pattern of the fragments of DNA. Here, there is the same exact pattern. So that means that O.J. Simpson's blood was in the crime scene. Check out the other types of blood. Let's see blood from Nicole Brown. She has kind of, this is very hard to see, but that's why people dedicate their entire lives to checking this out. She kind of has a pattern where right over here, the vast majority of fragments are. That matches a little bit of this pattern. So her blood was definitely in the crime scene. Second type of blood, it matches the pattern of the suspect O.J. Simpson. So this tells you that the 2 blood samples from the crime scene was from O.J. Simpson and 1 of the blood samples was from Nicole, the victim. Okay, so with this amount of evidence, at today's day, somebody gets charged for murder, okay, or for at least being in the crime scene. You can also use this to check paternity. So how can you use this to check paternity? You get a sample from mom, you get a sample from dad, from the son or daughter. Okay, and what you're going to see is that the son or daughter needs to have a pattern of restriction fragments or of these fragments of DNA. These are called restriction fragment length polymorphisms. Sorry. So we're going to see that later on, hopefully. Okay, but they're just these lengths, right, of these DNA segments that were cut. So the child needs to have a combination of pattern that comes from mom and comes from dad. Okay? So what can that look like? If the child has a little band right over here and mom has band right over here, that's not a problem. But if the child has a band right over here and mom has no band, but the father has one, then you can say that the combination between the mother and the father would give out that baby. If the father does not have any of the bands similar to the child, then you can say that the baby is not likely to be his. Okay? Okay, so polymerase chain reaction, these are PCRs. They're very similar to the idea of cloning genes, putting them in bacteria. The only thing that we're doing is we're not putting them in bacteria. All we're doing is we're using the polymerases, okay, DNA polymerase, to actually replicate a whole bunch of copies of a specific gene of interest or a specific fragment of interest. So what we do is we use primers. We use these very small segments of DNA that are going to attach to one side of the DNA of the strand. Okay, and we have another primer that's going to attach to the other side of the strand. So what that's going to do is, first of all, we're going to heat the cell up a whole bunch. Why do we heat it? So that we can separate both strands. We separate the strands with heat. And once we separate the strands with heat, we have this DNA polymerase, these primers that attach to each of these strands of the beginning and the end of sites that we want to code for. And once the primer attaches, we know that then the DNA polymerase can attach. And the DNA polymerase is going to attach and is going to start producing massively these replicates of this just segment that we want. So you're going to replicate the segment one after the other, after the other, after the other. Now, in order to ligate the segment or have the segment be bound, okay, we will decrease the temperature. So we increase the temperature to open up the segments, the 2 strands of DNA, so that each one of those can be replicated. And then we decrease the temperature so that the replicates can bind to one another and create a very strong segment. Okay, so we do these warming up, cooling down, warming up, cooling down cycles many times, because every single time that we are creating the cycle, we are creating new replicates. Okay, so in this cycle 2, for example, it yields 4 molecules, but in cycle 3 it's going to yield twice as much, 8 molecules, and we're going to do this for as many cycles as we can do it. So out of just, let's say that we only have one cell in our sample, it's only one cell big, and we need a whole bunch of that gene or of that DNA of that we're trying to analyze and that we're trying to target, what we can do is we can put that cell in this process through many, many cycles and end up with thousands of copies of that DNA sequence. Okay, so this is polymerase chain reaction. We can also clone. So all we do, this is actually super easy. We get the DNA from a somatic cell, that's just a body cell, and an egg cell, if it's the case of a vertebrate. Okay, so that egg cell, we take out the nucleus with its haploid sequence, okay, and we insert a diploid sequence of the somatic cell. And it's kind of like that egg cell was fertilized, right? Because now it's diploid. And then we insert all of this we do in vitro or outside of the organism, and then we insert this new egg cell that has the adequate amount of DNA into the organism so that it can grow and it can have its embryonic development and its fetal development in that organism, and then you simply have a clone. Okay? Okay, so this is just the big ideas of technology. Genes from virtually any organism can be cloned. Okay, super, super important. You can clone it with this plasmid transformation, putting plasmids into bacteria and just cloning them, just having them replicate, and in that way cloning your gene of interest or your DNA of interest. Or you can clone it with PCRs. So you can just amplify. PCRs are an amplification of a particular DNA sequence just based on this cycle of warming up, cooling down, warming up, cooling down, and having DNA polymerase replicate that DNA one after the other, one after the other. Okay, we can also test different fragments of length that you've cut down with restriction enzymes with electrophoresis. Okay, and you can do this with a whole bunch of different things. Forensics, you can test if a person has a disease, you can test if a person has a virus. We're going to be talking about the coronavirus soon and how you can test it actually with PCRs, okay, if you've got the virus or you don't have the virus, so we're going to talk about that soon. You can also create genetically modified organisms or organisms that are transgenic, trans meaning more than one organism, genic meaning genetics, so more than one organism's genetics in that organism, and you can clone animals as well. So what you guys are going to be doing what you guys were doing yesterday is watching these videos in Edpuzzle for CRISPR. CRISPR is super, super cool, and it's very similar to restriction enzymes, but even cooler, okay, because there's a specificity that you can do with CRISPR, that you can cut things that you want to cut. With restriction enzymes, it gets a little bit more complicated to know where to cut things and where to place things. CRISPR is super specific, so hopefully that helped. And yeah, watch this video, take some notes, and ask me questions in our discussion board if you have any. Thank you.